You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Future Tech Health podcast. I have Alessandra Sussulini, Assistant Professor of Analytical Chemistry at University of Campinas in Brazil. So, Alessandra, thank you for coming.
Thank you so much for having me.
I'm going to be talking about metabolomics. So, to start off, can you, what is metabolomics? What's a simple definition for people?
As a simple definition, metabolomics involves the study of the set of all metabolites in an organism. These metabolites are compounds with low molecular mass that are involved in the metabolism, such as amino acids, sugars, lipids, and so on. And metabolomics aims to measure these metabolites in an organism under a specific condition. For example, during a disease or a treatment, and many other aspects.
Okay, so my metabolomics, for instance, it's all the metabolites that my body produces when I consume food or breathe oxygen. Is that what it is?
Yes, metabolomics. The metabolome actually reflects what's going on in your organism. For example, if you have a disease, your metabolome will be affected. Or if you take any drug or consider also your diet, your metabolome will also change. It's like metabolome, we can say, is like a molecular phenotyping. It's just like a picture reflecting what's going on with you during a specific time and specific conditions.
Okay. Since a lot of biological processes are multi-step, how do you know if, the Krebs cycle, let's say, or, any cycle in my body? What if I have 5 steps to a process and I produce different metabolites at each step? Is all of that studyable? Is that my metabolome, or only the last compound that ends up being the final one?
Yes, metabolomics is a bit different from other omics, like transcriptomics, for example, because you cannot predict which metabolites will be produced from the genome, for example. So you cannot compute that because metabolites are end products from the cellular metabolism, but also they can act in different aspects, such as protein activity, and so on. So it is all around. So it's not just encrypted by the genes.
Well, if you're studying bacteria that's producing something, and you, first of all, how do you identify what metabolites an organism will produce? Literally, what are the lab? What do you do in the lab to study this stuff?
Yeah, when we talk about metabolomics, we are always comparing 2 conditions. So we can see, for example, if we compare a person with some disease with another person not disease. So we are going to compare their metabolites and see what's changing. So it is impossible to have, for example, the measurement of all metabolites in your body. What is important in metabolomics is to know which metabolites are changing due to a biological process. That's the main focus of metabolomics, these comparisons. So by doing these comparisons of these metabolite panels, we can see which metabolites are altered due to a biological process and then we can investigate the pathways where these metabolites are involved, and then we can make like some propositions about the mechanisms of this biological process going on.
But how do you study metabolites? Let's say you have E. coli bacteria and you want to study how it produces something or consumes some other, chemical or food. What do? Do you grind up the whole bacteria and, burn it up and put it through a gas chromatograph? Or how do you find out what it's producing?
So how to measure the metabolites, you want to know that?
Yes.
Okay. So you have many options to do that, to investigate this metabolome. The most used techniques are mass spectrometry, usually coupled to a chromatographic technique such as gas chromatography or liquid chromatography, and also nuclear magnetic resonance. So these are the main analytical techniques that are used to measure the metabolites in the most different samples.
Okay. If there's a process that goes on that produces, over, let's say over 20 seconds, a chemical process in an organism produces different types of metabolites as it progresses. Do you ever try to measure an organism, every second for 20 seconds, let's say, and see if the metabolites are changing at each second? Have you tried something like that? Is that even possible?
Is it possible? But usually in the normal metabolomics workflow, what we do is like a freeze time. For example, we do we call a metabolic quenching. So, for example, if we take a sample from a patient, like blood from a patient, first step is to freeze time. For example, like putting on extremely low temperature. So we stop the metabolism and we measure at that time point. So this is the conventional metabolomics workflow. It's not like a kinetics experiment. We just look at the metabolome at that time point, normally.
Okay. So what kind of research and work are you doing specifically? What are you working on right now?
Okay, right now my research group, we use metabolomics and also other omics techniques to investigate biomarkers for psychiatric diseases, more specifically bipolar disorder and depression.
What kind of biomarkers are associated with those disorders? Have you found any?
So we have found some interesting biomarkers. So we are in the phase of validating, but the most important molecules already connected to bipolar disorder, for example, are proteins related to the lipid metabolism and glycerophospholipids. So these are the main findings we have till now. Because bipolar disorder is a quite complex disease, so it's not an easy task to have it diagnosed, and the mechanisms of the progression of this disease is also very challenging. So we have found some clues about what's going on with a patient with this disease. So these clues are important because it can help the physicians to diagnose a patient, because usually the diagnosis for the psychiatric diseases is only clinical, right? So you don't have like a blood test for it. So that's interesting to have these markers to help the physicians to make a more precise diagnosis.
So what are some of the specific markers that you found, and what conditions do they appear to correlate with?
Sorry, can you repeat?
What biomarkers? What biomarkers have you found specifically, and what conditions do they appear to correlate with?
So the conditions, what do you mean about these conditions? About when I compare a patient with the disease with a control person?
Yeah, like if I have schizophrenia, are there specific biomarkers that you found for schizophrenics? Or if I just have depression, or if I'm psychotic, do you have, what specific biomarkers have you found, and for what conditions?
Okay, so most specifically for bipolar disorder, we have found these glycerophospholipids and proteins related to the lipid metabolism. So it's not only one biomarker we can define for each one of these diseases. So usually what we have are biomarker panels. So we can see different molecules that that are being altered due to the disease. So that's what we define as this biomarker panel. So we have some classes of molecules that are being altered, and now we need to validate these molecules we found to have more specific ones. So we have like a proper characterization of them. Because for exploratory studies, we first have this overview. We have this biological molecules, the classes of these biological molecules. And then we go further with the validation and the quantification of the biomarkers to define panels. So until now, we are doing these exploratory studies. We don't have definitive biomarkers yet because it is a very long process to have all these analyses and validations. So the next step after these validations, so we can say, okay, we found this, and that biomarker for each disease. So it is like a complex way going on.
But what does it tell you that you see phospholipids, in bipolar disorder? What is that hinting at? That's what's going on.
So we can see that the lipid metabolism is really affected by this disease. So many authors even suggest a dietary reposition of omega-3 or 6 fatty acids together with other lipids. So these are some studies made in the past, and these findings we are having confirm that there's a dysregulation in this lipid metabolism.
Oh, okay.
I don't know if I explained it properly.
Yeah, well, okay. So in people with bipolar disorder, it appears that they have an irregular lipid metabolism. So is it overactive or underactive, or are they producing lipids not commonly seen? Like, what are some specifics of what you're seeing?
So about specificity, I cannot tell you yet because we are in the validation phase. That's one issue. And usually what happens is that most of these lipids are down-regulated. So they have a decrease in their concentration, their relative concentration.
Any other conditions that you've looked at in addition to bipolar disorder?
We are now studying treatment-resistant depression. And for that, we are not only for the comparison of controls and diseases, but also we are doing some studies with alternative treatments with a psychedelic called Ayahuasca, because it is very interesting here in Brazil because it is like a beverage that's being used by many years for the people in the Amazon region. And now we have some interesting studies that are relating the use of ayahuasca for treating depression. Just after a single dose of ayahuasca, the patient the patient feels the antidepressant effects up for 14 days after the intake. So this is very interesting, and now we want to see it at the molecular level, what's going on, because we have the results from the psychologists. They are very exciting, and now we are using the metabolomics tools to understand what's going on with the biology of these patients taking ayahuasca as a potential antidepressant. So this is another field of study at my group right now.
Is ayahuasca effective for medication-resistant depression?
Yes. We have this study in collaboration with some researchers from the University of Rio Grande do Norte, here in Brazil. And they made this study already with the patients. These patients did not respond well to conventional medications. So these patients were selected, and a study was performed, and some patients received ayahuasca and other patients received placebo. And for the patients that received ayahuasca, they presented these antidepressant effects up for these 14 days after the intake of a single dose. So this is really interesting, and that's why we are employing metabolomics to understand better what's going on with these patients, because we know that's in terms of the psychological evaluation, they get better. But we want to see in terms of metabolism what's going on.
Any clues as to what is going on? Even though your data hasn't been validated yet, the reduced concentration of certain phospholipids in bipolar disorder, what does that tell you may be going on?
So this shows like a dysregulation in the organism. So that's why the person might develop the disease. So this is important. Why? Because it can help to have like a picture of what's going on, and the physician can say that if these lipids are dysregulated in their organism, so something can be predicted, like for the progression of the disease and so on. So I don't know if this is.
So how long of a path will it be to validation? How long will it take you to validate, and what do you think that you may find?
So this validation, yes, this validation step takes a lot of time because we need to find another sample sets from different places. So it is like a long-term study. We have been studying bipolar disorder in my group for 5 years already, and I believe we have still a lot of work ahead. So it's not so easy to determine this and validate the biomarkers. So initially we did these exploratory studies. We have now some clues of what's going on, some hints. So we are now going to focus on these lipids that appear to be more important to the disease, to the mechanism of the disease. So this takes a long time. It's like a work for more 5 years or even longer.
Were you surprised that you found, different phospholipids, different metabolites based on a condition such as bipolar disorder?
Yes, it is interesting because many people think that psychiatric disorder are just related to the psychological side, so they don't believe it has like molecular implications. So this is really important to show that the biochemistry of the body is completely changed with a psychiatric disease. So it's not just like psychological condition. So this is interesting. This is one point that is interesting to stress out in these studies. And these dysregulations, when we can find out where they are, so it's easier then to target new, like to point out new targets for drug development. So this is another interesting point. So when we see which pathways are altered due to a disease, so we can see what targets can be stressed out when developing new drugs, in which pathways these drugs may act.
Have you dealt with any of the microbiome? When you're looking at the metabolomics, have you ever found any strange results? And perhaps those results came from the microbiome of the area that you were sampling?
Yes, it might be due to the microbiome, or maybe due to diet or environmental factors. These are all things we need to consider when we are doing a metabolomics study, because we also have all these variables that might influence on the results. So that's why we need to work always with large sample sets, so we can try to avoid these variabilities, like the effect of these variabilities that should be minimal in comparison with the variabilities caused by the disease, for example.
Okay. So what the next few years are just going to be validating what you're doing, or are there new areas that you're working on?
Yeah, the new area is that one I mentioned before, for studying ayahuasca as an antidepressant. And this is the new study now in my group. And for bipolar disorder, we are still working on the validation. And of course, another interesting field we are exploring is to integrate multi-omics data. So we are now just acquiring data not only from metabolomics, but for other omics sciences such as proteomics and metabolomics. So we are now working on the bioinformatics fields to integrate this data and have, to have more definitions about what's going on with the diseases. So this is another field we are looking forward to work with.
Okay. With ayahuasca, what's the legal status of it in Brazil? Is it allowable to be used only by the Amazonian tribes, or is it usable by everybody?
It can be used by everybody in a ritual context. So, and also for research. So we have this law that allows any people, not only the tribes in Amazon, to use the ayahuasca in this ritual context. And for research is also legalized. So it is a very good opportunity for us in Brazil to work with ayahuasca.
Okay. Well, very good. What do you think is possible to figure out in the next couple of years? What are some of the big breakthroughs that you think will happen?
You mean for metabolomics in general?
Yeah, metabolomics in general and your research, yes.
Well, in terms of the research from my group, we are improving our exploratory studies. This is the first step. We are looking to work with this multi-omics strategies as well to improve the knowledge concerning the mechanisms of the diseases we are working with, especially bipolar disorder and depression, as I mentioned before. And in terms of metabolomics, an interesting thing that we are trying to work with is developing faster types of analysis, because it is not easy to have these mass spectrometries available, and analysis take a long time. And also the data treatment workflows, we are also working in improving to make it faster, and thinking that more people can have access to obtain these metabolomic profiles, and these metabolomic profiles can help the physicians to diagnose or predict diseases or propose treatments more effective based on the response of the patient, like in the context of personalized medicine. So this is what we think should be the future of this area.
How long does it take to figure out the metabolomics of a given chemical process? Is it a very difficult procedure? Does it take a while?
You mean to define a metabolomics study?
Is it expensive? Is it take a long time, or is it very quick? Like, what are the
No.
economics of it?
Okay, so metabolomics experiments are very expensive because they rely on mass spectrometry or NMR, so they are expensive analytical techniques. The analysis of each sample depends on what analytical platform you choose, but usually, let's say, about 20 minutes per sample. But for a whole metabolomics study, you need to have like a control condition, a test condition. You need to have all the quality controls of your analysis, so it takes many days to be finished. And the data treatment is also very challenging because it involves multivariate statistic tools, and for the identification of these metabolites is also very challenging because we rely on database searches. So it's not like a quick study right now. So that's why we are trying to develop other techniques, or simpler or cheaper, to access the metabolomics in an easiest way. So this might be the future, and this is what we are trying to test right now with techniques that are more simple, like for example infrared spectroscopy. So this is another field we are looking for. It's not like a detailed metabolomic profile that we can obtain with this technique, like for example with mass spectrometry, but for a general profiling, like a fingerprinting of a biological condition, it might be helpful. So this is another interesting thing to be studied in metabolomics.
Okay, well, very good. So what's the best way for people to get in touch with your lab or ask you questions?
So I have my email address, so it's easy to contact me. I can send you, well, I'm available to ask any further questions. I think email is the best way, the easiest way right now.
Okay. Well, very good. Well, Alessandra, thank you for coming on the podcast. I appreciate it.
Thank you so much for having me.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.